Welcome back, everyone. What we're going to do is go through some of these aims. We probably won't have time for all of them, but I'm going to try to catch a couple at the various levels of these aims. There were at least one or two groups on all aims. So if someone from any of the groups that had these aims wants to chime in, or anyone else, you are absolutely welcome to. So example 3 was study urinary tract infections. So what's some things that you want to say about this aim? Group 4, I know you're here.
I can chime in.
Go for it.
Well, no, essentially what Charlie is saying, it's just like that's a life's work, um, and not a very workable or testable hypothesis.
Absolutely. It's really broad. It's not a question. It's impossible to like. I can't say whether that's good or bad just because it's way too much. Thanks. And I am so sorry. I'm not actually able to see the chats well. So normally I'd be jumping all over that. But if you are putting in chat and I can't see it, I'm so sorry. Okay. Example 4. Perform RNA-seq on bladder tissue after UTI. Groups 2 and 5. I know you had this. What are some thoughts here?
Uh, we've mentioned that there are no specific timelines or conditions or model organisms mentioned. So basically, again, it's too broad, not very specific.
Yeah. Cool technique, great important group of urinary tract infections, but you're not actually telling me why you're doing it. So RNA-seq is a method. What question are you actually answering? Example 5, this is groups 3 and 6. Investigate all host pathogen interactions during urinary tract infection. All right, we got some more specifics. Do we like it, groups 3 and 6?
We said it was a bit too ambitious to be a singular aim.
Awesome. This scope is far too broad. So the words hypothetically are more specific, but all host pathogen interactions could mean immune, it could mean urothelial cell, it could mean other types of cells that are in the bladder, or all the pathogens that could infect the bladder. Um, Example 6, inflammation influences recurrent urinary tract infection. So now we're getting a little bit more specific here. What do you think, groups 4?
I was in that group, and I'm going to let the others take this if they want to.
If you prefer typing in the chat, feel free to, and someone else who can see the chat right now is welcome to read it out loud. Or feel free to chime in, or it's totally okay if it's a guess too.
I see the chat, so if someone puts anything in there, I'll let you know.
Thanks.
There's not really an indication of mechanism, if that makes sense, and also influences is really broad.
Yeah,
Um,
we were talking about how it could be a little more specific and directional in what it's trying to measure.
Excellent. Oh, sorry, there's another click there. Determine the role of inflammation in recurrent urinary tract infections. There are many inflammatory pathways, but it didn't provide specifics on what kind of inflammation might influence recurrent urinary tract infection or how. So which cells, which outcome. Again, even though we're getting some more specific words here, it's still a little bit too vague. All right, example 7, antibiotics alter the microbiome. Our aim was determine whether antibiotics alter the gut microbiome, urinary microbiome, immune response, epithelial repair, bacterial persistence, and recurrence. Groups 2 and 5. What do you think? This sounds like super specific.
It's a great question, but there are no approaches or specific techniques talked about, and it's too many microbiomes, so that's too overachieving.
Thank you. That's super, super important. Daspi, that's a great point. So this is like 12 aims together. It's awesome 12 aims, but it's too much, or okay, 5 or 6 maybe. And that's actually the reviewers will say this is overambitious, and it's not specific enough to any one of those types of aims. So sometimes too much is too much. And in this case, this would be an overambitious aim, as mentioned. Example 8, uropathogenic E. coli survives antibiotics inside bladder cells. The aim is determine whether intracellular bacterial communities promote recurrent infection. Groups 3 and 6, what are your thoughts here?
I think with this one, we were saying that like it would be helpful for it to have an approach and didn't have an approach in any way, or it had an approach of 16S sequencing, but I didn't explain why that was important or how exactly that experiment would like answer their aim and hypothesis.
Good. Tell me a little bit more. So there may be many people here who don't quite understand what 16S sequencing is. Are you able to tell me what 16S can do and what it can't do? Or anyone else in your group? Go ahead, Anna. I saw you on mute.
Oh, I can't. But that's I
Okay.
think what the
Okay.
problem is with at least for me personally.
Okay, great. Is there someone who can tell us, either in that group or otherwise, why, so that there's a nice aim, a nice hypothesis, and the approach is 16S, and the group is saying, hey, maybe this approach is disconnected with this aim. Can anyone tell me more thoughts on that?
It's organism identification, as far as I understand.
Good. It's organism identification. Is it like strain level identification?
I believe so, but I don't know enough to comment.
Totally fair. So I'll just give you a heads up. It is not strain level. It's more like genus level. I see something went in the chat. Often species or genus. Okay, thank you so much. You're right. So this approach is going to tell you who's there, almost, but it doesn't actually test this hypothesis about whether uropathogenic E. coli survives antibiotics inside bladder cells. So cool technique doesn't address the question. So this one's a little bit more subtle in how we're thinking about this aim in relation to this hypothesis and approach. All right, hypothesis. Urinary dysbiosis causes recurrent urinary tract infection. The aim is determine whether urinary dysbiosis causes recurrent urinary tract infection, and the approach is to compare the microbiomes of healthy women and women with recurrent UTIs, or urinary tract infections. So we've got some nice comparison groups. We've got a kind of specific aim. What are you thinking about this group 4?
Well, we kind of had causation, and then a comparison. And I think that with the approach needs to be tweaked a little bit to maybe like whether urinary dysbiosis precedes your recurrent UTIs or something. I don't know how to describe it in good terms of how to, you know, word that properly.
Hannah, you described that excellently. So correlation doesn't necessarily equal causation. So let's say healthy women and women with recurrent urinary tract infections do have different microbiomes. That may or may not be what is causing recurrent urinary tract infections. They may just have different recurrent urinary tract infections, perhaps due to the disease. So this one is, the problem is, can this observational study demonstrate causation? And the answer is no, it can't demonstrate it. So it can't make that direct connection with just this approach. So again, this aim is now part of a bigger picture. Example 10, neutrophils promote recurrent urinary tract infection by damaging the bladder epithelium. So neutrophils are a type of inflammatory cell, and the bladder epithelium is the cells lining the bladder. The aim is determine whether neutrophil infiltration promotes recurrent UTI. And the approach, I'm going to measure neutrophil numbers. So I've got some really specific things I'm doing. I'm talking about a specific inflammatory cell. What do you think groups 2 and 5? Thanks for being here, Anna.
They're not.
Overall, our group,
Sorry.
sorry. Overall, our group thought it was pretty good. The hypothesis was pretty clear. The aim was pretty good. It's more so the approach that we wish we had a little more information. So like measuring the neutrophil numbers, that's good, but how are you planning on doing that?
Absolutely. So there's actually some reasonable thoughts going on here, but the approach lets us down a little in directly testing that hypothesis. So again, just knowing whether there's more or less neutrophils doesn't tell you if neutrophil inflammation is directly promoting recurrent UTI, or maybe it's just happening in the presence of recurrent UTI. So you're right, Madison, and the groups that explored this having some more specifics on this approach might still be able to address this hypothesis, but as is, it does not. Biofilms protect bacteria from antibiotics. The aim is determine whether catheter biofilms increase treatment failure. The approach is confocal microscopy. Super cool. What do you think, groups 3 and 6?
So we thought that this is a like a very broad aim, as well as they don't mention what type of which bacteria they will be using to make the biofilms, as well as what type of antibiotics they will be using, uh, or antibiotic treat. They don't talk about how the antibiotic treatment for these biofilms and how they can know whether the antibiotics are working or not just from the confocal microscopy.
Good. So again, really cool approach, but does it actually test the hypothesis? So like you said, Hrishikesh, microscopy alone can't demonstrate treatment failure. It can say, hey, I can see this thing or I can't see this thing, but you will actually need some kind of bacterial survival data to directly address this aim and hypothesis. Example 12, the urinary microbiome influences susceptibility to recurrent infection. And my aim is determine if the urinary microbiome regulates susceptibility to recurrent UTI. And I'm going to treat mice with antibiotics. What do you think group 4?
I did like the hypothesis and aim, but the approach is so broad. Um, like what is it measuring? Infection, recurrence, bacterial burden, inflammatory response, or it needs to be more specific in that regard.
Great. So there's several things that could have been improved on here. So there are things that are missing, particularly with regards to the approach. It doesn't say exactly what does it mean when we say the urinary microbiome. So are we measuring some level of diversity? Are we looking for specific types of microbes? There's no recolonization or mechanistic link here. It just says treat mice with antibiotics, and it doesn't say what about the urinary microbiome do we think might be influencing susceptibility and how might it influence susceptibility. So some nicer words here, but the scientific logic doesn't totally connect. Example 13, commensal bladder bacteria. So commensal just means not pathogenic or microbes that might live in a healthy bladder. Um, Commensal bladder bacteria suppress uropathogenic E. coli colonization by competing for iron. So we've got a specific hypothesis that we're looking at. The aim is determine whether commensal bladder bacteria limit UPEC or uropathogenic E. coli colonization through iron competition. My approach is competition, assays, iron supplementation, and mouse infections. Groups 2 and 5, what do you think?
I think they should narrow down on which commensal bladder bacteria they're talking about, like it's either lactobacillus or bifidobacterium or something like that, because that will influence the kind of experiments that we run a lot, because different bacteria, they grow in different like media and they have different nutrition requirements and things like that. So I feel like this had to be a little more specific.
Great. And this is a particularly important point coming from Ujjani. So you heard some of the graduate students who introduced themselves, maybe focusing an entire PhD on something like E. faecalis or lactobacillus, because there's so much to explore within a single type of bacteria. That would be a really good point in thinking about this question. So what some of the problems with this aim is are we only using one commensal strain? Which one? How many? Are there alternative mechanisms? So let's say you combine a commensal bacteria with a UPEC, and sure enough, it decreases the UPEC. Uh, could there be anything else besides iron competition that might be driving that? Is there a way for us to prove that it's iron supplementation or iron competition? And are we actually measuring iron competition? So we say that we're going to supplement with iron, but we don't say that we're going to measure it. So thinking about this as a good aim, there's some other more additional details that would be really valuable in this project description generally. Okay, example 14, groups 3 and 6. You had this. Do you want to give some thoughts on example 14?
This one was very strong. Like it mentions about everything what they are gonna do. Like they also have like expected outcomes, and it like aligns properly to I think so what actual specific aim should be.
Excellent. And just so you know, example 14 did not say strong on the Google Doc. So these groups identified that this was a really scientifically logical set of aims, hypotheses, approach. So here, determine whether intracellular bacterial communities promote recurrent urinary tract infection by protecting uropathogenic E. coli from antibiotic treatment. And the hypothesis was that these intracellular bacterial communities do act as protected reservoirs to promote survival against antibiotics. And the approach tested this hypothesis fully. And here they also provided an expected outcome and impact. So these studies will establish whether intracellular bacterial communities represent a mechanistic driver of recurrent urinary tract infections and the impact. So the bigger picture, this work could identify new therapeutic strategies to prevent recurrent urinary tract infections. So now you've gone through a lot of really common problems that are seen with AIMS, as well as some strengths that are seen with AIMS. And so, what I'm going to have you spend about 5 minutes on now, this is a short amount of time, but you're going to try this out. I'm going to have you consider a topic that you are or may be interested in studying and develop your own NIH style aim, hypothesis, and approach. Now, the most important is aim here. So if you don't get to hypothesis or approach, that's okay. And this says 10 minutes, but I'm actually only going to give you 5. Um, so give this a shot. And if you are at a point where you're just not quite able to do that, that's okay. You can still be a contributor by listening to other aims because I'll have you guys go back into breakout groups momentarily. But what I'm going to have you do right now is you're just going to you can turn your camera off, I'll turn all of our sounds off, and I'm going to put on a 5-minute timer, and you're just going to try to write an aim. Welcome back. We are rounding out our hour, and we don't have a ton of time to debrief this, so I'm going to just comment. Thank you for letting me join your groups. Thanks for participating. Sometimes it can feel really different to try to write something than critique it, and it can be a totally different sense of like, whoa, I know how to critique, but now I have to like apply all that knowledge. So I hope you got a little bit of that experience and a little taste of aim writing and some good comments on from your rooms as far as like this was
Yeah.
confusing or this was really clear. Final thoughts for today from me before I turn it back over to the group to Caribou is there's just a checklist. So as you go into your own aim writing for various things, remember one biological question in one sentence. That's an aim. Is there one central hypothesis? Do your experiments test that hypothesis? If successful, what new knowledge will be gained? Is the scope realistic for one aim in that budget period? And would a reviewer understand what success looks like? I'll round it out there and wish you all happy aim writing in your futures. And it was delightful to be here with you. And I'm sorry we kind of ran out a little bit of time, but I'll turn it back over to Caribou.